Hello, welcome to my presentation. Let me continue introduce my new project work about evolutionary models. In the last presentation, I introduced how to simulate DNA sequence data. In this speech, I will quickly review the background and goals of this project. Then I will show some representative results from the simulation data. Later, I will focus on the latest achievements in recent months, including the complete analysis of DNA sequences and how to capture the most appropriate model recommended according to Bayesian rules in the analysis log file, and how to count the number of recommended models and generate a correlation matrix. And finally, how to use the correlation matrix to visualize the results. Biogenetics is a very popular direction at present, and the phylogenetic tree is an important tool to study the biological genetics. With the improvement of computing power, it is possible to analyze large orders of DNA sequences. To generate a phylogenetic tree, the most important thing is the selection of evolutionary models, because the maximum likelihood method is a relatively mature statistical method for parameter estimation and has a good statistical theoretical basis. When the sample size is large, the maximum likelihood method can obtain the minimum variance of parameter statistics. Therefore, as long as a reasonable and correct alternative model is used, the evolutionary model based on the maximum likelihood method is considered to generate good evolutionary tree results. The evolutionary model used to build the phylogenetic tree in our project is also based on the maximum likelihood method. This evolutionary model requires an appropriate substitution model for analysis. So the quality of the substitution model will directly affect the quality of the final phylogenetic tree. The reason why the phylogenetic tree is sensitive to the selection of substitution model is the composition of DNA sequences. As can be seen from the left picture, DNA usually exists in a double helix structure. After magnification, it can be observed that DNA is actually composed of 4 basic elements, which can be expressed as A, C, G, and T. Because the units of two DNA strands at the same position have one-to-one correspondence, we usually only need to pay attention to one of them when analyzing data, which can be regarded as a DNA sequence. The DNA difference of different organisms is reflected by the order of ACGT in each position. In biology, when A and G exchange or C and T exchange, this is called a transition. And if A and C are exchanged or G and T are exchanged, this is called transversion. Because the substitution model needs to set the rate of change between each DNA element, and the rate of transition and transversion in reality is found to have certain rules after measurement. It is often used as a reference for setting the parameters of the substitution model. Now let's take a closer look at the 22 substitution models available on IQTree. As can be seen from the right picture, the parameters of the substitution model are divided into 2 parts. The first part is represented by the connection diagram on the model name, where lines with the same color represent the same set parameters, and the type of colors represent uh, the same set parameters. Out the And the types of color represent the numbers of parameters to be set. The second part is represented by the histogram on the right of the model name. 4 columns of the same length represent that the 4 elements have the same frequency in the sequence, while 4 gray columns of different lengths represent that the true frequencies of the elements in the sequence needs to be set. For example, in the Jus-Cantor model, all connections are black. That is, there is only one parameter to be set, and the probability of all DNA elements transforming each other is equal. The equal height of all gray columns means that the 4 elements are equally frequent. For example, in the GTR model, the connection of 6 columns means that 6 different rates should be set. The unique height of 4 gray columns means that the element statistics of the sequence to be analyzed should be carried out, and the real data after statistics should be taken as parameters. The figure on the left reflects the same information, but it only counts the number of parameters to be set and explains the naming rules. Now that we understand the composition of the parameters of the substitution model, what I want to discuss next is the goal of this project. That is, whether the 22 replacement models with different tendencies are too crowded. If the answer is yes, which models are redundant and why? If the answer is no, what irreplaceable characteristics do this model have? The current research focuses on the selection of evolutionary models rather than considering whether existing models are redundant. The purpose of this project is to find the advantages and disadvantages of the model through the comparison of redundancy and try to find out the future development of evolutionary models. Next, I will introduce the specific implementation method of this project. It can be divided into 5 parts. The first part is to simulate the DNA sequence data, which I completed in the task last trimester. According to the preset 22 models, 2,200 DNA sequence data files with 16 taxa and 5,000 lengths were generated through 100 for loop. The second step is to use the model finder function of IQ-tree to analyze each file and give the most appropriate model name. There are also many other tools available for model choosing. The reason why IQ-tree is used here is that IQ-tree was also used in the first step when simulating data. Since the standard file naming format is set when generating these files, it will be easy to analyze these DNA sequences with IQ-tree in the second step. The third step is to locate and extract the model name from the log file generated by IQ-tree after analyzing the sequences. Files are usually text. You can locate the keyword first and then get the location of the model name. After the capture is completed, the model name and the data of each parameter will be combined into a new line of data and stored in the CSV file. In this way, we will finally get a 2,200 line CSV file. The details will be described in the code part later. The fourth step is to count the CSV files generated in the previous step. Calculate the occurrences of each model and establish a correlation matrix with the model used to simulate data. The fifth step is to visualize the correlation matrix on a two-dimensional coordinate axis with 22 model names in both horizontal and vertical coordinates. If all goes well, we will expect to get information about whether the model is redundant from the generated visual images. The code here shows part of my achievement last trimester. It includes how to import a necessary library and dataset, and setting the correct relevant parameters, and how to use the correct loop to ensure the generation of simulation data with the required quantity and characteristics. I won't spend too much time explaining the code for this part. If you are interested, you can watch the video I made last trimester. Next, I will focus on the ideas and the methods I have written down after discussing with my teachers and groupmates this trimester. As shown in the figure, in the part of using IQ tree to analyzing data, I have retained some relevant parameter settings and even for loop usage method used when simulating data. The reason is that each file name needs to be obtained when using IQ tree to analyze data. Therefore, to complete the analysis, I only need to change the last command executed in the system to the model find function. In the next part of capturing the model name, I still retain the code for parameter settings and for loops. It should be noted that due to the special mechanism of IQ tree, during the process of model finder, IQ tree was not only tested the 22 models mentioned previously, but also tested the 22 model variants on this basis. Therefore, it is very likely that the result will have a model with plus F or plus I or plus blah blah. We need to take an extra step to remove these noises. This is to ensure that no error occurs in the next step of statistics. The good news is that after removing these noises, all the results will fall into 22 model names. The next part is the establishment and visualization of correlation matrix. First, generate a matrix of 22 times 22, and then prepare an additional function that can translate the model name into a number. This matrix represents each model used to simulate DNA sequence data that is finally chosen by the model finder function. Next method is to read the CSV file line by line, add one to the corresponding position of the model in each line, and divide the matrix by cardinal number, that is the number of unwrap. To get a final matrix in our language, there is a function that is very convenient for outputting correlation matrix images. That is bitmap. As long as the correct matrix is input, you can get an image that reflects the correlation by color depth. Once we have completed the whole project progress and output the correct image, the next step is to analyze the image and have a conclusion on whether the model is redundant. But after our group discussion, we found that the three parameters involved in this project may also affect the correctness of the conclusions. Therefore, we need to repeat the project and use different parameters to see the impact of these parameters on the result image. These three parameters are the length of DNA sequence, which we call it sequence, and the number of taxa, and the number of for loops. First, we test the number of for loops. From the graph, we can see that when unwrapped, which is the for loops numbers, become significantly larger. A small number of models show more possibilities. But from a macro perspective, unwrap does not have a great impact on the results. Next, I compared the effect of DNA sequence length on the result image. The same as the number of for loops, we also get the conclusion that DNA sequence length has little effect on the results. But when it comes to testing the impact of taxa on the result, we found that when taxa is set very high, like 64, the coincidence rate of the model chosen by model finder for the model used to simulate data is significantly improved. Therefore, in the final step, we need to discuss the classification of different taxa. Finally, I would like to discuss some conclusions from this project. The first one is to compare the use different taxa sequence and the for loop times. It can be seen that Druscanto F81, K2P, and HKY are considered as models with low redundancy, and TNE, TN, TPM2, TPM2U, TPM3, and TPM3U are considered as model with medium redundancy. K3P, K3PU, TRME, TRM, TRM2, and blah, 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 and GTR are considered as models with high redundancy. What can be found is that when more parameters are involved in the substitution model, the DNA sequence files generated by the model simulation are more likely to be recommended to other models. This is a preliminary analysis, and other comparison and analysis are in progress. The details will be recorded in this Prometheus report. I hope the research results of this project can help us better understand the role of substitution models and evolutionary models in building phylogenetic trees in the future, and even become a better tool for studying biological genetics. Well, that's all of my presentation. Thank you all for listening. Bye bye.